Hello everyone, today we will discuss about proteins. Proteins are one of the mega nutrients. All the proteins, they have simple basic molecular unit in their structure. We call it as amino acid or a monomer. These monomers, they combine together to make more complicated structures. We call them as proteins. For example, these are two amino acids. When they come close and make a chemical bond between them by a chemical reaction, we call it as dipeptide. When more than two amino acids are chemically attached together, we call it as polypeptide. Proteins are either made up of one polypeptide chain or more than one polypeptide chains. There are 20 amino acids that are present, and these 20 amino acids, they combine in a different way to make different proteins, as proteins are really specific in their function because of their structure. Let's see a basic structure of amino acid. Amino acid has a central carbon atom that is attached with carboxyl group on one side and amine or amino group on the other side. This is our variable group and this is hydrogen. So amino acids are different from each other only because of this group. Our group make different amino acids, different from each other. Let's see how polypeptides are made. Polypeptides are formed by condensation reaction. So what is condensation again? Condensation is a chemical reaction in which two molecules, they come close together in a way that a new chemical bond is formed and a molecule of water is released. So proteins, these are made by this reaction. Let's see. This is one amino acid. This is another amino acid. Then number one amino acid, number two. When these two come close together, the carboxyl group and hydrogen of amine group react together and a water molecule is released. When a water molecule is released, a new chemical bond is formed between carbon of one amino acid and nitrogen of another amino acid. This is the bond. We call it as a peptide bond. And this complete molecule is now, we call it as dipeptide. And this is happened because of condensation, because in this process a molecule of water is released. Let's discuss about structure of protein. Proteins are very complicated in their structure. To understand, we will divide these in 4 levels. Primary structure of protein, secondary structure of protein, tertiary, and quaternary. So primary structure, it just has straight chain with a simple sequence of amino acid between them. So this primary structure just has amino acids in a straight chain. But when we talk about secondary structure, this straight chain doesn't remain straight anymore, because hydrogen bonds are formed between different molecules in a chain. So hydrogen bonds, they result in a either coiled alpha helix or a folded beta plated sheet. So hydrogen bonds that are established between different molecules, they automatically make this straight chain either coiled structure or beta plated sheet. So let's have a look about tertiary structure. Here the coils or the folds that are made in tertiary structure, these chains are further coiled and further folded. And here were only hydrogen bonds that were present in different molecules. In tertiary structure, more hydrogen molecules are formed between different parts of the chain. Now, ionic bonds are established between different charges, positive and negative charges on different molecules, and also disulfide bridges are formed whenever two molecules of amino acid cysteine come close together. So the sulfur from one molecule, it bonds with the sulfur of another amino acid. So disulfide bridges are also made in tertiary structure. So for the proteins that are made up of only single polypeptide chain, so the tertiary structure defines their final 3D structure. So this is only for the proteins that are made up of only one single polypeptide chain. Let's see quaternary structure. In quaternary structure, this is especially for those proteins which are made up of more than one chains. So this is not about single polypeptide chain. These quaternary structures are for those proteins which are made up of more than one polypeptide chains. And those proteins, they have quaternary structure as their 3D final structure. For example, insulin, hemoglobin, and collagen. These polypeptides, because these are more than one polypeptides, and these chains are connected to each other with cross links, and often these are more compact and tightly bonded. For example, in the case of enzymes, these are little bit spherical because of the compact arrangement of the folds of the polypeptide chains. Let's see functions of proteins. For example, enzymes, these are proteins. These are roughly spherical. And this is because of the polypeptides that are tightly bonded. Enzymes, they have role in metabolism. Some enzymes are involved in the breakdown of large molecule into smaller molecules. So enzyme help them in the breakdown. In some other cases, enzymes are involved in the manufacture of the big molecules. They are also involved in the breakdown of molecules as in digestion, and these are also involved in the making of the large molecules, for example, hormones or proteins, other proteins. The second type of protein, antibodies. Antibodies, they have their role in immune system. They have two long and two short polypeptides that are present in them, and these antibodies have special regions. Their amino acid sequence vary greatly. And if we talk about third type of protein, that is channel protein. As we know the structure of plasma membrane, so plasma membrane, it controls what enters or leaves the cell. So all the substances, they can't enter into the cell. So these special channels that are protein channels, actually these are proteins. So these channels help for those substances that otherwise can't pass through this membrane. So these channels help to pass these molecules to inside of the cell. And in these channels, there are different types of amino acids. Some are hydrophilic and some are hydrophobic. So because of those amino acids, this protein is just turned up into a tube shape, and that is working accordingly. Let's talk about structural proteins. Structural proteins are strong, and these are made up of long, straight polypeptide chains that are held parallel to each other, and these parallel polypeptides are linked to each other with these bonds. These have cross links between them that make them very strong. For example, keratin, that is present in our nails and hair, and also collagen, that is present in the connective tissue.